The evolution of the first life on Earth is a complex and fascinating process that spans billions of years. While the exact origins of life remain uncertain, scientists have proposed several theories and gathered evidence to understand how life may have emerged. Here is a brief explanation of the leading hypothesis known as abiogenesis. Early Earth conditions. The early Earth had a hot and volatile environment. It was bombarded by meteorites and experienced intense volcanic activity, resulting in a constantly changing landscape. The atmosphere primarily consisted of gases like methane, ammonia, water vapor, and carbon dioxide, lacking oxygen. Primordial soup. In the 1950s, the famous Miller-Urey experiment demonstrated that simple organic molecules, including amino acids, could be produced under conditions similar to those of the early Earth. This experiment supported the idea that the building blocks of life could have arisen spontaneously through chemical reactions in the oceans. Protocells. Protocells are hypothesized to be the precursors to cells, exhibiting some characteristics of life. These early structures likely consisted of a membrane-like boundary that enclosed self-replicating molecules. The protocell membrane protected the internal components, allowing for the segregation of the protocell from its environment. RNA world. The RNA world hypothesis proposes that RNA played a central role in early life. RNA can store genetic information and catalyze chemical reactions, making it a prime candidate for the first genetic material and enzymes. It is believed that early self-replicating RNA molecules, known as ribozymes, facilitated the synthesis of essential biomolecules. Evolution of metabolism. As protocells became more complex, they likely developed primitive metabolic pathways. These pathways allowed for the acquisition and utilization of energy sources from the environment. The availability of energy would have provided a selective advantage, driving the evolution of more efficient metabolic processes. Development of genetic code. The genetic code, which enables the translation of genetic information into proteins, likely evolved from the interactions between RNA molecules. Through natural selection, specific RNA sequences and their corresponding amino acids could have become linked, forming a primitive code. This early genetic code eventually evolved to include more sophisticated mechanisms for protein synthesis. Transition to cellular life. Over time, protocells with improved genetic and metabolic capabilities would have outcompeted their counterparts. These more advanced protocells eventually gave rise to the first cellular life forms. The transition to cellular life involved the development of more complex cellular structures and the evolution of mechanisms for efficient reproduction and growth. It's important to note that the precise details of these processes are still being explored and refined by ongoing scientific research. New discoveries and insights continually contribute to our understanding of how the first life forms may have emerged on Earth.